Does it have anything to do with the recycling of the endosome? So even if it is fuses with the membrane, so they don't remain functional. Okay, so that's what it means. So for achieving functionality, see, as such, it's not only about recognition, it's about you recognize these elements and then induce all these downstream events and to produce type 1 interferons and NF-kappa B activation and so on and so forth. And that is why you get the effector function associated with the innate immune recognition intracellularly. Okay, so that is why you have membrane associated TLRs, and then you also have the intracellularly present innate immune receptors like TLRs, some of those. And obviously, then you have node-like receptors. You also have Rigai pathway, which actually is cytoplasmic in origin. So its element actually are present in the cytosol. Some of these things are also present on mitochondria. For example, MAVAS is another type of innate immune response present in mitochondria. But these were discovered very recently, so some more work need to be done, but very critical for antiviral defense. Activated T cells vary depending on antibody. Does the phenotype of activated T cells vary depending on the antibody due to which it was activated? If yes, then no. Well, say it again. So can you switch on your video and or audio and because somehow this is not making sense. Bardula, ask your question.
So can you hear me?
Yeah, I can.
So does the phenotype actually change depending on the antigen due to which it is activated? Like for one specific disease, the phenotype of the activated T cell will be different compared to the other.
Well, see, as there are three things. We are going to talk about the activation of T cells, but what happens is there are three signals which are very critical for activating your T cells. Okay, the first thing has to be the ligation of T cell receptor. And what does TCR recognize? The peptide present on MSC class 1 or class 2, right? So that's signal 1. Signal 2 is the co-stimulatory molecules, and signal 3 is the cytokines present in the microenvironment. So once you have these three signals, so then you get activation of your T cells. Now, depending on this third signal, what is the composition of the microenvironment? What kind of cytokines are predominantly present? So that will decide whether T cells will acquire one phenotype or the other phenotype. Okay, for example, you have Th1, you have Th17, you have Th2. So if we talk about the helper cells, okay, and for cytotoxic, obviously the phenotype actually is you can call them as TC1 or TC2 cells, but predominantly their function actually remains to kill the target cells. Okay, target cell means the cells which are infected and they are recognized by these activated T cells through these peptides displayed by MHC molecule, right? This third signal actually is responsible for providing a particular phenotype on T cells.
Sir, so this third phenotype will also be responsible for memory?
Yeah, obviously. So once cells are they differentiate from naive to effector, right? And of these effector cells, after carrying out their function, so almost 90-95% of these cells die. And you're left with 5% of those cells, right? And which will become memory cells. Same is the case with all kind of phenotype which have been generated, whether it's Th1, whether it's Th17, whether it's Ts2, or cytotoxic T cells. Okay, the phenotype of these cells remains same, but this is not to say that phenotype will not change subsequently when these cells are reactivated or are recalled in the response. Again, at that time, whatever the cytokines are produced and are present in the microenvironment will decide what phenotype it will acquire then. Okay. And this phenomena is called plasticity. Okay, so for example, at one time point you generated Th1 cells, and of those Th1 cells, they became effector cells and some into memory, right? Now this Th1 cell, depending on what microenvironment they are in, can also become Ts2. Okay, so this is a very changing phenomena. That and what decides this change? The microenvironment, the composition of cytokines. Okay, so it's not fixed. And at molecular level, basically this is carried out by the epigenetic changes that you have in your um, the genetic material in the nucleus.
Fine, sir.
Yeah.
Sir, apart from this, is the TLR heterodimers, which you showed in one of the figure,
Yeah.
are those the only possible combinations, or these are just known combinations till now?
Well, these are some of the known combination, but it's not like that every single TLR will interact with every other TLR. So this has not been discovered. Maybe depending on the microbial infection or the ligand present, so they might, but this is not true entirely that every single TLR will interact with every other TLR, either in the endosome or on membrane of these cells. Whatever is known, okay, is known as of now, and it's been quite long time when these TLRs were discovered. So if more interactions were to happen, so they must have been discovered by this time. Okay.
Sir, can you just go back to that slide once again, if possible?
This one.
The TLR thing, which has this one, sir. So here, if you see in plasma membrane, there are heterodimers, but if you see the internal compartments, they are just single TLR. There are no dimers, if you see. So is there any specific reason for that?
Whatever is discovered, right? Rather than TLR4, for example, you don't have a heterodimer. So there are other elements also, right? For example, MD2 is also involved in TLR4 signaling. So there is another membrane associated molecule that we call as MD2. And once you have this interaction of TLR4 and MD2, so it recognizes LPS. Okay. In case of TLR2 and 6, these heterodimers do play a role, and same is the case with TLR1 and 2. Okay. Now if you think about it philosophically, so the element which is recognized by these TLRs, so for example, if you have peptidoglycan, so what is the composition of that peptidoglycan, and whether this is also associated with some moieties coming from, for example, TLR4. So then you have to activate those which can recognize peptidoglycan, and you also have to activate TLR4, for example. So the complexity of ligand is going to stimulate signaling through the set of TLRs, right? And for the TLR which are present intracellularly. So as I said, these predominantly recognize the genetic material of microbe. However, they don't recognize your own genetic material, okay? And they don't have to, because it's present in the nucleus, right? And usually you don't find this species of DNA or RNA which can actually come in contact with these TLRs. And probably this is to ensure that you do not activate these cells all the time because of stimulation through your cellular genetic material. Okay, so this gets recruited only when you have something entered inside these cells from outside, like these microbes.
Fine, sir.
Okay, so anyone else has question or clarification on any of the things that we covered?
So,
Yes, sir.
so I have a short clarification. So regarding the dendritic cells, so you had talked about DC's sort of interact with adaptive immune system. So why can't we say that along with NK cells and NK T cells and gamma delta cells, DC also comes under the category? Like, can we put superficially?
Dendritic cells you want to put under what?
The
At
category
inter-
molecules which are acting at the interface of innate and adaptive.
well, rather, actually, dendritic cells are the one which actually connect innate and adaptive immune responses. And for the simple reasons that they take up antigen, they release initial set of cytokines and take up antigen and process those, and in so doing, they also activate your T cells. Yes, you can call them that these cells also act at the interface, but you don't really call it as interface, you call them as the connecting link. Okay, connecting link between innate and adaptive immune responses. But these other cells, like gamma delta T cells, NK cells, they are not connecting link as such. Okay, so they don't basically take up antigen and then activate the cells of the adaptive immune response. But cell-to-cell interaction does take place. But this is not through the peptide MHC and TCR. This is not occurring in that way. It's just that they are activated, they start spitting different cytokines very quickly after infection. And they have really very less, you can say, repertoire of the receptors they express on them, and they only recognize certain ligands. Okay, so in that way we talked about whether these cells are working at the interface or as we just have talked about, that dendritic cells, macrophages, and also B cells, so they actually link innate and adaptive responses. B cells predominantly during the recall of the memory phase. Okay, because initially you don't have so many B cells which express that particular B cell receptor so that they can actually take up this antigen. But during memory phase, so you find more number of these B cells which can recognize this antigen and eventually can serve as APCs. But not during the initial phase of the response, the acute phase of the response.
Fine, sir. Thank you.
Yeah.
So
Yeah.
I have some general queries. So
Mhm.
let's say if I have inflammation at my hand and if I
Mhm.
rub that, then it increases further, right?
Yeah.
Why it happens so?
Well, um, okay. So anyone want to answer that? Well, yeah. So I don't know the answer. Okay, but it could very well be that if you have this inflammation and then basically you provide another insult. Okay. Um, now, one thing is that you are not rubbing it with a very sterile hand. Okay, so that means you might actually introduce more microbes in that area. That could be one. Second thing is that you also have many of these pain receptors. And if you actually rub it, so you activate those pain receptors, and that also basically uh, promote inflammation, right? Um, you can actually also subside the inflammation. For example, if you press it so that the accumulated fluid, it get disrupted, rather dispersed. Okay. That actually depend on what kind of inflammation you had, whether actually you got a bruise or there's internal inflammation, or it's just the accumulation of this fluid, maybe for example under the skin, right? So all those things are going to decide whether you promote or you dampen this inflammatory response. Okay, but there are many more factors which probably might be responsible for playing a role. As I said, for example, if you press it with a dirty hand, and usually it's not like you press it only with the other hand once you have basically uh, sanitized it with alcohol or something, right? So it doesn't happen that way. So you try to basically rub it just like that, right? And invariably on your hand or on your skin, so you have lots and lots of microbes present, and because of additional microbes that you are providing, so you might increase the level of inflammation.
Okay, sir. So, and second inquiry, sir, that we know that memory cells remains for a longer interval of time.
Yeah.
So, do they maintain themselves or the same set of cells remain throughout the lifetime?
Yeah, they maintain themselves, and this is through a slow turnover rates. Okay, so and they require certain growth factors for their maintenance and also this turnover. Actually, memory cells are also having this stem cell like property. Okay, the side of, yeah,
So they divide, yeah.
yes, they divide but very slowly, unlike effector cells. Okay, effector cells divide very rapidly. Okay, within the 24 hours you might get rather 3-4 division within 24 hours. And if you have hundreds and thousands or millions of those cells, you can imagine how many of these cells will be present within 24 hours, right? And 48 hours. So this division is very, very rapid, very fast for effector cells, but for memory cells, this is actually a very slow turnover. The cytokines which are responsible for maintaining and responsible for this turnover of memory cells are different as you find for the effector cells. Okay, for example, for effector cells, IL-2 is very critical. But for memory cells, IL-15 and IL-7 are critical for maintaining their numbers and slow division rates.
Okay, sir. So one more question, sir. if I get first vaccine and first vaccine of Covaxin and second dose of Covishield, then what will happen?
Nothing.
It's fine. I mean,
Yeah, well,
that will be more good, or as compared to this, the one which we are right now taking 2 doses of the same vaccine.
well, I think, in my opinion, from immunological perspective, this is always better. So that you get first dose with the whole virus and second dose with only a part of it. For example, in Covishield, you have the spike protein only, right? In Covaxin, so you have the whole virus. Yes, this is inactivated, and inactivated vaccines, they have their own problems, particularly for coronaviruses. So inactivated vaccines have not been shown very effective, theoretically, rather even for historical perspective. Whenever they used inactivated vaccines, so these inactivated vaccines did not work that efficiently. And actually, if you want to read more about it, there was a recent article in Nature where they showed that some of these vaccines which are heavily used for managing coronavirus, so they are going to be actually devastating. Okay, because somehow they don't induce the kind of response which is protective. For example, you have this Sinovac, which is also an inactivated vaccine. You also have this Covaxin, which is also an inactivated vaccine, right? And there are quite a few. And these vaccines somehow are not very good in inducing response which are durable, which are lasting, and which provide you a long-term protection. This is not to say that only spike carrying vaccines are also great, okay, because there are mutations in spike. And it was actually shown in very recent articles that the antibody response which was induced following vaccination or even during the infection, okay, so that somehow fails to neutralize these mutants, okay. Omicron was not neutralized by the antibody response which was induced following these vaccines and even from the convalescent sera. But one also has to understand that you don't completely rely on antibodies, right? So you also induce your T cell responses. And for T cell responses, so not many epitopes are changed from one strain to the other strain. So still your T cells are functional and they can actually protect you. But problem with T cell protection is they don't prevent infection. They control it well. Okay, once you're infected, the infection is controlled faster by the activity of T cells, particularly the memory T cells. Okay, but antibodies actually can prevent infection, and we know that that antibodies somehow with coronavirus are not very protective. Okay, protective means they don't prevent infection. So even if you got this this vaccine or both doses or even the booster dose, so chances are that you may still get infection, but that will be controlled because of the activity of your T cells. Okay, don't be under the impression that if you got this vaccine, so you are safe, you will never get infection. You will still get infection, and that is why. Well, I got infection recently, and I had both of these doses. But I don't know whether this was corona or common cold, because I didn't get tested. Okay, And everybody is getting these infections. Some are underreported. Some are not getting it tested because within 2-3 days you recover, right? So, but my thinking about this whole thing is that everybody can get infected again despite vaccination. Okay, and people keep on saying that this new strain is mild. Yes, it is mild because you have immunity, particularly mediated by your T cells. And those T cells can control once you are infected with this virus, this new strain, within 2-3 days. Earlier, when you are not immune, so then the course of disease was actually much longer. Okay, so you don't rely only on one branch of your immune system for this virus. So you have antibody and you also have T cell response. And we more or less know now that antibodies somehow do not protect you. But your T cells come into your rescue to make it a milder form. Okay.
Yes, sir. So one last query, sir.
Yeah.
So thymus is not present in adults. Then how T cells proliferate in adults?
Well, this is not to say that thymus is not present in adults. Okay, so yes, there is thymic atrophy, and it shrinks if you grow older. But you also have these lymph nodes, right, where you already have formed your T cells, and we did mention that these T cells keep on changing their footprints. So if they were of one type, now they can become a different type depending on the microenvironment, the composition of cytokines. Okay, and that somehow helps you keep going. Okay. Yes, novel specificities are not generated. Whatever you already have, you just have to work with those specificities. Okay, but the maximum time where your T cell responses, T cell repertoire is formed is when you are, let's say, up to 13, 14 years old. After that, you don't generate new specificities.
Okay, sir.
Well, they keep on changing from one phenotype to the other phenotype, depending on the cytokine present. And that is responsible for inducing protection.
Okay, sir. Thank you.
All right. Yeah. Anyone else has a question? Okay. So if not, let's do.
So,
Yeah.
so when you say that antibodies are better than T cell response in the vaccine scenario, so how is it decided that which vaccine for the disease, which vaccine should have antibodies or which vaccine should have T cell? Like HIV virus has T cell vaccines and the other ones have antibody.
Well, one general comment about this whole scenario is for RNA viruses, antibodies are not so great. Okay, this is not to say that you generated good antibody response, but problem is with the virus itself. Virus keeps on changing. And if it keeps on changing, okay, so whatever the antibody response you have generated, so now this is not protective anymore. Okay, because you already have modified these epitopes which need to be neutralized. Okay, but these alterations do not happen in T cells. because T cells actually recognize these sequences of peptides, 7 amino acid, 8 amino acid, 9 amino acid, right? And it's not like that you are going to change each one of those, even if you have mutation occurring. Okay. Now, question is, if your antibodies are not protecting with a new variant against a new variant, so if you are not being provided with the protection, but you have the T cell responses induced. Okay, and T cell responses will be induced only for or more efficiently for attenuated vaccines, not with the killed vaccines. Okay, with the killed vaccines, so you actually tend to generate only antibody response. Okay, and that is why I don't like Covaxin to begin with. I don't like it, and it's not my personal liking or disliking. It's from immunological perspective. Okay, so in the very beginning I said that this vaccine is going to be a disaster. And why this Covishield is better? Because it gets into your cells. Okay, it does not replicate, but it gets into your cells. And once this adenovirus gets into your cells, so this will be processed in a way that you can actually generate decent T cell responses for both helper and cytotoxic T cells. Obviously, you also generate antibody response. These other vaccines, like you have one from Moderna and another from Pfizer BioNTech, which actually were never used in India. So they are even better. And why they are even better? Because one thing is they are mRNA based vaccines, and this actually can also stimulate your innate immune responses efficiently. And so basically then you got your innate responses, you got your antibody responses, and you also got your T cell responses. They are much better than the vaccines that we have here. Out of the vaccine that we have here, so Covishield is still better. Okay, but I think that was not your decision which vaccine you will get. So you went to this hospital or this vaccination center, and whatever the vaccines they have, they gave you, right? So probably they never asked for your preference, which vaccine would you take. But something is better than nothing, right? But
So,
as I said, for viruses which mutate so frequently, antibody responses are not very protective, and you actually rely on T cell responses, not to prevent infection, but to control it efficiently. Yeah.
so for the control of infection, for bacteria as well, we can have T cell responses, for example, for mycobacteria.
Yeah. Yeah, because Mycobacterium is also an intercellular pathogen. Other bacteria which are not intercellular, so you can actually rely on antibodies. Okay. Yeah.
And sir, a general
Yeah.
in the pandemic scenario, we usually say that we should stay away from asymptomatic people because they don't show symptoms. But if they don't show symptoms, and we at the same time we also say that fever is an immune response of the body against the infection. So are we actually saying that the asymptomatic people have higher immunity because they don't show any immune response, such as
These are actually very relevant questions, but not everybody is an immunologist, right? And they keep on saying whatever they want to say. There are actually 1,000 reasons to explain or to say one thing or the other. Okay, some are scientifically true, and some are basically based on just these statements that people keep on making. Okay, now if somebody is saying asymptomatic, I don't believe this. Okay, so it's impossible to get your body manifesting symptoms if you are infected. Okay, the intensity of these symptoms might be milder. Okay, and milder in the sense they can be so that you cannot really assess that there are significant physiological changes in your body. Okay, and then you start calling them as asymptomatic. This is not to say that maybe once in a while you did not have this feverish or the normal physiology disturbed, behavioral changes. So all these things are going to happen if virus has gotten into your system. So if somebody is saying asymptomatic, I don't believe it. Okay, it cannot be that virus is in your system and you are asymptomatic. It's impossible to have such a scenario. The second question is, who develops these unrecognizable symptoms versus the symptoms which actually can easily be recognized by your own body as well as anybody else. One of the region or one of the thing could be the amount of virus dose you have received. Okay, so for example, rather actually we wrote a couple of commentaries about this COVID-19, saying that if you are infected with a low dose, it's actually good, because then you can actually trigger your immune system and you have some level of protection. Okay, but if you get a lot of it, your body is not able to control it so efficiently, and then you start getting disease, and many a time this can be even worse. Okay, so what essentially that means is why this mask and all those things are good, because even if you get infected, so you will get infected with a very, very low doses, which can simply be considered as actually a way of immunization with the live virus. Okay. However, if you are not taking all these precautions and you get infected, you don't know how your body is going to respond. A lot of people did die. Okay. Yes. So you need to keep taking these precautions. You have to stay away from people who are actually secreting virus. And even if you say that asymptomatic, the virus actually is secreted in the very early stages of infection. Okay, not later on when you actually are symptomatic. So in the very early few days, 2 or 3 days, virus actually is produced by an infected person. Okay, but when you actually have all kind of symptoms, so this is because of your immunopathological response. At that time, you don't get too much virus produced in these individuals. So this early time point is very critical to be away from that infected patient. Yes.
Okay, sir, fine.
And I see on campus, there are so many students who even are not using these masks. वो चाय भी पीते हैं और वो सारा सब कुछ काम करते हैं अब वहीं पे. And I'm actually pretty sure now everybody on campus is infected. Everybody. There's not even a single person who is not infected with this new strain. This is a bold statement, but entirely true. Because this is actually very infectious virus. Whether this is manifested in symptoms which are recognizable or not is something which can be debated. Okay. But it's not like that you get these emails that these 3 students got infected. Some other faculties got infected, some staff members got infected. No. Well, to tell you the truth, everybody got infected. And if some have not, they will get infected very soon. Okay. And if you want to not get infected, so keep using these precautions. People can say it's mild, but let me tell you, it's not mild. It's mild to the extent that it's not going to kill you, but it's going to do every other thing. Anyways, so let's start with a few of these things. Okay, so components of lymphatic system. So we did talk about this slide. So you have lymph, which is actually the tissue fluid, which enters into your lymphatic vessels. And then you have lymphatic vessels, which can be divided into 4 types. The smallest one are capillaries, lymphatic capillaries, and the largest one is lymphatic ducts. And lymphatic vessels and lymphatic trunk, they fall in between. Lymphatic organs. So you have thymus, lymph nodes, spleen, and tonsils. And then you have these cells. And we broadly talked about these cells as part of your myeloid and lymphoid lineage. Okay. Now, if you talk about lymph node, okay, so these were the one, actually, these nodules like structure, which are in between these traversing lymphatics. Okay, these are the lymphatics, and in between you have these nodule-like structures. Okay, these are basically the lymph node. Okay, so they are they are actually small, round, or oval-shaped structures. They are located along the pathways of your lymph vessels. Their length can vary from 1 to 25 millimeters. Typically found in clusters. Okay, and they receive lymph from different parts of body, and which part of body they drain to. Actually, this is based on the location. Okay, for example, if you have these lymph nodes in hand, so the lymph which actually is flowing in your hand, so that will be drained through these lymph nodes. They are found individually throughout your body tissues. Okay. Now, if you talk about the structure of these lymph node, so actually which is shown here. Okay, this is actually a pretty well organized tissue. Okay, the outer layer actually is the you have basically, you can actually divide this into cortex and medulla. Okay, cortex is on the outside. Okay, so this actually is cortex, and this greenish shaped structure actually is the sinus capsule. Okay, underneath you have the subcapsular sinus, and these actually are organized in these different areas. And what divides this into these different areas is trabeculae. Okay, so you have this trabeculae which divide them into, for example, these distinct areas. Okay, all right, the part which is closer to the capsule is called cortex. Okay, so this actually part is cortex. And then you have the paracortex area. So this actually is the paracortex area. Okay. And medulla is basically in the middle of this. Okay, so this is medulla. Medulla, paracortex, cortex. Now, these organized structures that you have within this lymph node, okay, so we call them as germinal center or germinal center in the follicles. Okay, so this is follicle and these are the germinal center. So these are the sites where your B cells respond to antigens and in germinal center, right? Paracortex is the area where your T cells meet antigen presenting cells. Okay, so paracortex is the area where actually you activate your T cells, right? Now, so there are also some blood vessels here or lymphatics. Okay, blood vessel is for supplying blood to the lymph node, right? And lymphatics for, there are two types of lymphatics that you have. One is afferent. So these are afferent. They basically feed these APCs carrying antigen, or the antigen enters through afferent lymphatics. Okay, afferent. And efferent lymphatics are the site through which activated T cells exit. Exit. Efferent. Okay, so efferent vessels are the sites or these vessels through which your activated T cells leave lymph node and they then start circulating and get to infected tissues, right? So APCs, they come loaded with these antigens through afferent lymphatics, through these, right? And when these APCs come, they actually somehow find their place to sit in somewhere in this area where T cells are located, and they sit here and they keep on screening these T cells depending on what antigen specific T cell receptor these cells are expressing. And as soon as they actually match the TCR with the peptide presented by these APCs, those T cells start getting activation. Okay. Is that clear? The structure of lymph node. Okay, and you know that these are present actually throughout your body, right? They are present everywhere.
Could you repeat the function of germinal center?
Germinal center are the sites where your B cells get activated and they start making antibodies. Okay. Because these, as you can see, physically these are actually present in these areas where antigen can come. And if these B cells, they don't have to actually engage with APCs for initially producing these antibodies, right? Antigen come through these afferent lymphatic. And when B cells see the presence of that antigen, so you start getting activated your B cells, and these B cells will start producing antibodies, right? And those antibodies that they can actually then be circulating, they are found actually in your blood. Yes?
Fine, sir.
Yeah. Okay, so spleen. Spleen basically is something like this. Okay. Location. So this is present in the epigastric region, and gastrum actually is your stomach. Okay. They are actually present on top of the stomach in abdominal cavity, right? Location of spleen. For example, if you have directly inject something in spleen, or if you have to take a biopsy of spleen, so you just pierce between 9th to 11th rib, and you can actually directly reach spleen, right? So these are all those ribs. So you can count that this is 9th and 11th, and if, within this area, so your spleen is located on top of the stomach. Okay, this is the largest lymphatic organ in the body. Okay. It can vary considerably in size and weight depending on the stage. Stage means, for example, during infection, the spleen is enlarged actually quite significantly. Okay. This is also called as the graveyard. Okay, so whatever the stuff that you have, all these microbes, they are predominantly can be found in spleen, and you induce these responses in spleen. The structure is again organized pretty well. Okay, so this is the basically structure of the spleen. You have this trabecula, which separated in different regions. Okay, you have some part that we call as red pulp, where it has a lot of these RBCs, and other part is white pulp, where you have your lymphocytes. Okay. On the outer boundary, basically this is the capsule, as you can see here. Okay, pretty well organized structure. Thymus, which is actually the primary lymphoid organ, right? You have this heart, and on top of this heart, so you have this thymus. Okay. And you already know that these are the lungs. So very close to all these things, you have the thymus. It's lobulated structure. You have two lobes. Pretty elongated structures. It's a large organ in the fetus, and as you keep on growing old and old, so there is a regression of thymus. This is also called as thymic atrophy. It's the site where your T cells develop. Okay. Initially, they start. These T cells actually are present in bone marrow, and from bone marrow, so they get to thymus. And in thymus, they decide whether these cells will make it to periphery or they die out. Okay, whether you make CD4 T cells or you make CD8 T cells, all those processes are decided in thymus. Okay. Okay, it occupies the thoracic cavity behind sternum. Okay, thoracic cavity. Produce this molecule that is called lymphopoietin. It's a growth factor for your T cells. Okay. Again, the structure is also same. So you have capsule, you have trabecula, right? Also pretty well organized structure. So it's divided into cortex, medulla, and there are different lobules in it. Okay. As I said, so as you grow older, so there is contraction of thymus. Okay. So what is this? Yeah. Well, you have different types of cells in thymus. Some cells actually make contact with lymphocytes and help their survival. These are called as nurse cells. Okay, they're present in this thymic capsule. Thymus is the site where actually you can find a lot of apoptosis happening. Okay, almost 95% of these cells in thymus undergo apoptosis. Very few cells make it to periphery to become either CD4 or CD8 T cells. And the simple reason for this is because you have to only let those cells get out of thymus which are not reactive to your own antigen, but they recognize foreign antigen, right? Now, a question arises, how come these self-antigens, specific T cells are eliminated, and how foreign antigen specific cells are selected? Okay, so this process actually is decided by positive and negative selection of your thymocytes. So we'll talk about that. There is some question here. Oh, okay. Let's stop it. Yeah, let's stop it. So I didn't realize that we are running out of time. Okay, leave. So attend your next class. So I'm just stopping it.